Hi there. In the second video on photosynthesis, we'll be talking about the two photosystems, which are referred to as the Z scheme, and why they're needed to achieve the plant's metabolic needs. We'll then talk about how plants can regulate electron flux through either photosystem 2 or photosystem 1 to adjust the relative amounts of ATP production, which is primarily energy, which versus NADPH, which is high energy electrons for biosynthesis. And then we'll conclude with a talk about these very important reactions in the Calvin cycle in which carbon is fixed, carbon dioxide. And this is the source of almost all organic materials on the planet. These carbon fixation reactions are also referred to as the dark reactions since no sunlight is needed for these processes. They do occur during daytime. Now, here we see on an energetic scale the two photosystems. Photosystem 2, where the process starts, and photosystem 1, which starts at a higher energy and ends up at a higher energy. The approximate differences are very closely matched here, and that's because they're both absorbing visible photons. The energy of a photon is dependent on its wavelength. Visible sunlight is what hits the Earth's surface, and so that's what's available. So the visible photons are being absorbed here. The same energy visible photons are being absorbed here. This just starts off at a higher energetic level than photosystem 2 does. So the requirement of two photons is in part because of the amazing property that photosystem 2 is able to rip electrons out of water. It extracts them, producing oxygen. This is the source of oxygen on the planet, and protons. If you extract the starting electrons from such a stable source as water, which is down here, and you have visible energy, visible photons to promote an electron, this is about the maximum energetic level you can get to, but you have to produce a molecule called NADPH, which is related to NADH. We'll talk a little bit more about the difference. But even though this is ever so slightly higher in energy than NADPH, it does not have sufficient excess energy to affect this transformation all by itself. If it did, it's conceivable that plants could get by with one photosystem. But instead what it does, it's going to transfer electrons down the electron transport chain to lower energies, and using that, photosystem 2 to pump protons that will be used for ATP synthesis. Now, since it's at a higher energy level, a second photon comes in, an electron transfer process will occur, and this has sufficient energy to reduce NADP+, creating this high energy molecule which is used for biosynthesis, the combination of small organic molecules into larger organic molecules. So functionally, that's different from what we saw with NADH, where most of the electrons are donated to the mitochondrial electron transport chain. This overall scheme is referred to as the Z scheme, although I admit when drawn this way it looks more like the letter N. And on this next slide, we're just really emphasizing here that for visible light, you only have a certain amount of energy. If you had ultraviolet photons, and the way we're going, we may soon have them, you might have enough energy to get by with a single photon. So now in the Z scheme, with a photon being absorbed here, I should have stressed, a photon is absorbed here, and a photon is absorbed here. So to complete the Z scheme, it's a two-photon process. If you pursue that, you have a fixed ratio of ATP production to NADPH production. But there are times when the cell might need either more ATP or more NADPH. So the plants have evolved a short circuit, if you will, using a protein called ferredoxin to only shuttle electrons in photosystem 1. Remember, photosystem normally goes to the production of NADPH, but instead, in this short circuit, the electron is instead donated to ferredoxin, which donates the electron back to a plastoquinone. Now you're getting proton pumping from photosystem 1. Photosystem 2, which was right about here, was also donating electron to plastoquinone and doing uh, proton pumping. So this is a way of increasing the relative amounts of ATP relative to NADPH. Now switching gears to the dark reaction, here you can see what's happening. We have carbon dioxide and a 5-carbon sugar, and we are going to produce 6 organic carbons, 2 3-carbon sugars of 3-phosphoglycerate. The enzyme that catalyzes this reaction is called ribulose bisphosphate carboxylase, or Rubisco for short. People have speculated that Rubisco is the most abundant protein by mass on planet Earth, which is perhaps a pretty good choice as it's the ultimate source of all our organic molecules being created from inorganic carbon dioxide. Now here is the Calvin cycle, and this is the most complex cycle or metabolic network we've seen yet. I won't do the stoichiometry because the production of a single molecule of glyceraldehyde 3-phosphate requires many turns of the cycle, but in a net we have 3 molecules of input. Let's consider 1 molecule of input. We'll have 1 5-carbon sugar, again 5 carbon, 1 carbon, 6 carbons. We're producing 2 3-carbon molecules right here, but these are required for the cycle. These are not being siphoned off as sugars for generic use. And the energetics are summarized here. We're going to invest 9 molecules of ATP and 6 molecules of NADPH to produce a single molecule of glyceraldehyde 3-phosphate. Now in this slide I'd just like to remind that, which we saw in the evolution lecture, that the accumulation of oxygen occurs about, I would say, about 1 billion years after photosynthesis develops the ability to split water, releasing oxygen. This is the reaction that is the source of all the oxygen, and for 1 billion years oxygen was being produced. It was being absorbed into the sea in the form of carbonates and various iron adducts. So basically the Earth was an oxygen sponge for 1 billion years. Finally, that sponge became saturated. All the buffering capacity was used up, and then oxygen began to accumulate in the environment. Now here I'd like to draw the very clear analogy between oxidative phosphorylation that occurs in the mitochondria versus photosynthesis, which occurs in the chloroplast. We have high energy electrons here in the form of NADH, which were generated from the digestion of foodstuffs. The electrons are passed down an electron transport chain. A proton gradient is established by proton pumping, and this proton gradient is used to drive ATP synthesis. And while it's not as essential for the comparison, the organic molecules are oxidized to carbon dioxide. Oxygen is consumed as an electron acceptor, producing water. In photosynthesis, we have the high energy electrons that are produced based on the absorption of a photon. It undergoes a series of downhill electron energy transfers to power proton pumping, forming a proton gradient, which will drive the synthesis of ATP. Then we have a second photon, which promotes an electron again to a higher level, and that creates the molecule NADPH, which, as we saw, played a critical role along with ATP molecules and carbon dioxide in the carbon fixation cycle, producing carbohydrate molecules. And again, oxygen is the byproduct of the water splitting reaction, this ultimate source of the electrons, which are being energetically promoted. So then I end with this slide that we saw in the lectures of oxidative phosphorylation. As you prepare for the midterms and review your knowledge on these topics, this slide will be very useful for you to gauge your understanding. There's a little bit of almost everything we talk about in here. Okay, thanks for listening.